Hello, my name is Mary Couture, and I am a sophomore in the Randall Research Scholars Program, majoring in mathematics and microbiology. I'm working with Dr. Yuping Bao in the Department of Chemical and Biological Engineering, and my project is the evaluation of mitochondrial variation in different breast cancer cell lines. The mitochondria is the powerhouse of the cell, until it isn't. In a healthy cell, the majority of the energy production in the cell is performed in the mitochondria, where ATP, the energy currency of the cell, is synthesized. Because of this incredibly specific function, the mitochondria in a healthy cell has an incredibly specific characteristic jelly bean shape. However, in a cancer cell, the mitochondria does not undergo the standard means of production of ATP because it is dividing incredibly quickly. Because of this lack of use of the mitochondria, we hypothesize that the mitochondria will in fact change in shape in cancerous cell lines. This is an example of the means of production of energy in the mitochondria of a healthy cell. It produces ATP through what is known as the TCA cycle, which is a highly efficient process for making energy from glucose. However, this process does take time. In a healthy cell, that isn't a problem. In a cancerous cell, however, the cell is dividing too quickly to be able to rely on the mitochondria, and thus it undergoes a process known as aerobic glycolysis, in which a smaller amount of ATP is produced along with a larger quantity of byproducts. This aerobic glycolysis, however, is much quicker, and so it makes it easier for the cell to continue dividing at the extreme rate at which cancer cells usually divide. This leads us to the question, what are the differences between mitochondria in healthy cells and those in cancerous cells? We know that aerobic glycolysis is occurring in the cancer cell because of what is known as the Warburg effect, in which we can tell that the production of lactate from aerobic glycolysis is affecting the environment around the tumor. We also know that the mitochondria themselves are underutilized because the means of production of ATP requires a specific membrane gradient within the mitochondria of pH potential. This potential gradient is not maintained correctly in the mitochondria of cancer cells. To look at this question, we are examining three cell lines, the first of which, MCF10A, are simply healthy breast tissue cells. These will give us a baseline for what the mitochondria of healthy cells look like. The next cell line we are looking at are MCF7s, which are weakly metastatic cells. They form tumors that spread to other parts of the body, but do not do so particularly aggressively. Our final cell line, MDAMB231s, which I will simply refer to as 231s, are an incredibly aggressive metastatic cell line. By examining these three cell lines, we hope to discover some of the changes in the mitochondria of cells as a function of the metastaticity of the cancer. This will hopefully allow us to determine the role of mitochondria in metastaticity and thus begin means of prevention of the development of more metastatic tumors from more benign ones. In order to determine the changes in the mitochondria, we are first going to determine the changes in the shape, as form following function is an incredibly important part of biology. The first test that we are doing is known as dynamic light scattering, and it simply determines the size of the mitochondria that we have isolated. The second test, scanning electron microscopy, allows us to form a 3D visualization of the surface of the mitochondria and thus visualize their shape. And the third test, transmission electron microscopy, allows us to visualize the interior structures of the mitochondria by taking incredibly thin slices of the preserved samples. In order to look at the mitochondria at all, however, we must first isolate them from a whole cell through an isolation protocol. In this protocol, whole cells are swollen in a hypotonic buffer so that their membranes may be ruptured in a homogenizer. Once they have been ruptured, they are then centrifuged, first to remove any remaining whole cells as well as large nuclei from the homogenate, and a second time to separate out the mitochondria from the rest of the supernatant, which will then be discarded. Once the mitochondria have been isolated, we then perform dynamic light scattering on them in order to ensure that we have in fact isolated the mitochondria as desired. This example of a dynamic light scattering performed on a protocol on 231s has two peaks, one around 450 nanometers and another around 1100 nanometers. The literature value for mitochondrial size is around 1000 nanometers, which means that while we do seem to have mitochondria in this sample, they are unfortunately in a contaminated isolation. So the isolation protocol needs further refinement. Once the presence of mitochondria has been confirmed, the mitochondria can then be fixed and imaged using either scanning electron microscopy or transmission electron microscopy. In scanning electron microscopy, the sample is preserved and then coated in metal so that a beam of electrons may be scattered off of it. This allows us to see the three-dimensional structure of the mitochondria. In transmission electron microscopy, however, the mitochondria are preserved in resin and then incredibly thin slices are taken. An electron beam is passed through this sample. Incredibly electron-dense regions will reflect the electrons back, whereas electron-poor regions will allow the beam to pass through. This creates an image with dark areas where there is a significant quantity of matter and light areas where there is very little. This allows us to visualize the interior structure of the mitochondria, as opposed to simply seeing the surface. Moving forwards, we will take a functional protocol and characterize all three cell lines. This will allow us to determine the changes in shape and interior construction as a function of metastaticity, as well as allow us to characterize the number of mitochondria present in a given number of cells for each cell line. Once we have this information, we will then move on to a functional analysis, first using a confocal microscope to determine the leakage across the mitochondrial membrane, which is what gives rise to the changes in membrane potential. We will then take a protein assay and determine the function of different proteins within the mitochondria that are involved in the TCA cycle. This will tell us where in the cycle the mitochondria is not functioning properly, specifically complex 1 and 2. I am now open for questions.
So do you have any idea how the changes in the mitochondria are contributing to the metastatic potential of these cells?
This is described through the Warburg effect, but the changes in the mitochondria change the function of the cytoplasm and also mean that certain genes change the function of proteins within the cytoplasm, and also mean that certain genes that should be activated are not being activated correctly, and genes that are used to help convert, say, pyruvate to lactate in the aerobic glycolysis cycle are activated when they should not necessarily be. This is putting the cells into an extreme stress response and is thus, we hypothesize, leading to the continuing and is increasing the metastatic behavior of these cells.
Why do you use both an SEM and a TEM when you image the mitochondria?
Because information simply about the external structure of the mitochondria or the internal structure on its own would not be enough. We need both pieces of information to determine the changes in shape of the mitochondria as a function of the mitochondrial membrane, as well as whether the proteins involved in the assays would be in the correct positions or even be able to anchor at all.
What changes in structure would you expect to see in cancerous mitochondria?
So the first thing that we would expect to see would in fact show up on the scanning electron microscope as a change in the shape of the mitochondria themselves. The surface to volume ratio is incredibly important within biology, and so a change in that surface to volume ratio would indicate that the protons, as part of the membrane potential that are supposed to be moving through the mitochondrial membrane, are not doing so correctly, as well as the ingredients involved in the TCA cycle. So that would be the first thing we would expect to see, would be a change from that jelly bean structure to either a more rod-like or most likely a more spherical shape, indicating a lack of functionality. Further, we could expect to see some degradation of the structures within the mitochondria, which is what the TEM would allow us to see, indicating that the anchoring of the proteins themselves and the orientation of the TCA cycle was not correct.